We're going over the notes from yesterday. If you missed something, get it down, and we're going to draw lots of pictures to help you. Okay, so on the Ed puzzle, what did they say are the two types of transport?
Active transport.
All right, good. Transport. Okay, whose job is it to transport? What cell structure are we talking about that we drew? Cell membrane, right? Not cell wall, but cell membrane. All right, so you said passive, and we wrote all this stuff yesterday. So passive and active. Okay, which one uses ATP, energy?
Active.
Right, so active ATP. So that's where...
It's called adrenaline. Hold on, wait. I forgot it again.
It's okay. Adenosine triphosphate. But you don't need to know that yet until Monday. We'll talk about it.
Right.
To move molecules, which direction?
So from high concentration to low.
For active?
Right. No, from low to high.
Opposite, right. So if they want to go one way, you're saying, oh, no, we're going to push you the other way. So from low to high. Now, we wrote all this yesterday. All right. What do we say passive does?
It goes with the flow.
Yeah, go with the flow.
All right. Go with flow.
What's that?
I'm going to end up beating this thing up.
Why is it messing up on us?
Oh, flow.
My bad, that's all I'm in.
See, oh, you can't realign it. Those can you? No.
Dang.
I used to love to realign.
I used to realign too.
Like that.
Hey, Teron.
Okay. So somebody last period said just go with it. And go with it is basically, we said is diffusion, right? And osmosis. Now, which of those moves water only? Only water. Osmosis. All right.
So you're going to let us know when you start to do it?
So this is water, yeah. And then this is other small molecules. Oxygen, CO2, small salts, right? Amino acids.
So those are the things that just go.
Glucose. I mean, anything that's, tiny, but water is H2O osmosis. So with your colored pencil, if you did not emphasize that, I would. So O. That's how I remembered it in 10th grade. Okay. So any way to keep it straight, because we know you're going to have a question about that. So then we drew a picture. So what picture did we draw first?
A
juicy grape.
Yes.
Oh, right.
Okay. All right. And then we drew a slug, right? Did we do the slug next?
Yeah.
All right. Did we label all the slug?
Yeah.
Did everybody get it?
No.
No, I didn't get it.
All right. So let's draw the slug, and then we'll start the new pictures. All right. So the slug. So here he is. And usually when I say, okay, this juicy slug has a lot of what in its body?
Protein.
We know there's protein,
But it's water.
but there's a ton of water. So we're going to put a whole bunch of water. So take the blue and let's put every dot is going to be, I bet 1,000 water molecules. Yeah, there's just going to be tons and tons and tons. All right, so that's always going to represent water for us. All right, salt. This thing is eating plants and being a nuisance, so we're going to put a whole bunch of salt on top of it. Now, there's salts in your body, there's salts in the slug's body, so that's why I put a few of them in there.
What happened why do people be putting salt on the slug?
Okay, so you're going to be able to answer that question in just a second.
They're trying to kill it.
All right, so we put salt. Now, where do you have a high concentration of salt? Outside or inside?
Outside.
Outside.
Outside. All right. So which way is it going to go with the flow? From high to low. So which way is the salt going to move?
Down. I mean to the right. I mean inside.
Inside. Yeah, inside. So I'm going to draw an arrow and write salt. This is where, if you can picture and identify which way one of the things are going to move. So the solutes are going to go in. What's going to be pulled out in the opposite direction every time?
Water.
Yes. Water goes out. Now, what did you just do to that slug? What's the word that we say when you run out of water, you lose water? Dehydrate. So because you dehydrate it so fast, it's like someone has melted, because I'll hear students will say, What causes it to melt? Well, what's happening is it's pulling the water out so fast that the cells are not maintaining homeostasis. They can't pump water back in, or at least not fast enough. So it's going to look like this little shriveled up, looking very sad and dead slug. Okay?
How fast would it like take to go from that to that?
Seconds.
Well, I wouldn't say seconds, but I would say probably minutes. No, maybe not totally shriveled. It'll just look like a mess of nasty, but it'll look pretty shriveled up. So let's think about an earthworm. We don't even have to put salt on an earthworm. Take a fishing worm and put it out in the hot sun.
And then it's
They're not gonna dry up.
Why?
The water evaporates out this sun.
So water moves from the cells out by what process? What do we say is always water moving?
Osmosis.
Osmosis. So this is osmosis, and diffusion is the salt moving. All right. So here's my new examples. We'll just put the date out to the side. So let's draw what happens at the grocery store. This is why they put water. They keep spritzing the vegetables with water. Okay. All right. So let's do. So this is going to be a cell of a piece of lettuce. All right. So here's lettuce cell. All right. So if you work for the grocery store, and they are the produce department, they're going to say that you have to spritz, or you have to have those little sprayers on the vegetables. Mm-hmm. So if you don't, well, first of all, when you bring them in, they're nice and crunchy and they're fresh. What's this big, huge organelle called inside of a plant cell? What's stored inside the central vacuole? Water. Okay. These are crunchy, fresh vegetables. They don't look brown. They aren't slimy. They are crunchy. That is any fresh vegetable or fruit. So if I just say, okay, I'm just going to leave it out there and I'm not going to spread any water on it, what's the cell going to look like?
It's going to shrivel.
Right. But you didn't add heat to it. All you do is just leave it alone. So what's the central vacuole going to look like?
It's going to look all
It's gonna be empty.
Yeah, it's gonna be so dried up that everything kind of pulls in like this. Now you've got a nasty piece of lettuce that's limp, but let's say it's still green. It's not slimy yet. What do you think we can add to this?
Water.
Yes. So if we add water to it,
you can still save it.
You can still save it. And then it goes back like that. So that's straight up osmosis. We didn't add any sugar or salt to it. We didn't add any ranch dressing to it. All we did was put it out there on the counter or just not spritz the vegetables. So that's lost money for that store if they don't keep their vegetables and fruit fresh enough. So that's that. All right, so that's one example. Does that make sense a little bit more? Water. Okay, so water left, and I would probably need to put that. So if you have a blue, I'm going to say water left by osmosis, and then if we add water, it's going to fill back up. Okay, so osmosis. All right, the next one is red blood cells. So we're going to draw that whole scenario yesterday when they said that if you are given fresh, pure water in your veins, it would kill you. Okay, so we're going to see why. Uh-huh. So in here is distilled water. This is the word that they're going to use on your exam. Distilled water is pure water. Okay, it has zero anything in it. It's just plain old water. Okay.
Where can you find that at?
It is, you just buy distilled water, and then we have actually a machine here that is a distillery, so we can actually make it.
Oh, so
It's kind of like a water filter in a way, you know, that passes and filters all the junk out, and then you're just left with plain old water like that. All right, so let's put a red blood cell. So we prick your finger. If we did this under the microscope, we put plain distilled water on the slide. We prick your finger and drop blood. Let's draw a red blood cell in here. Looks like a little donut without a hole. And what does your blood taste like other than iron? Like metal?
Blood.
Yeah.
But what else does it taste like? It's like metal and
Nothing.
yes, salty.
Yeah.
So
How?
we're gonna put some dots inside, and those dots is like the salt because they naturally your blood is a little salty. Well, that's because they're all perfectly balanced inside there. So now, tell me what's gonna happen. Where's there more water? Pure water is only water. There's going to be a ton of it on the outside. So now tell me what's going to happen.
The water's gonna move in.
By what process?
Osmosis.
Very good. So water comes in. The rule is if one thing goes one way, the other thing goes the opposite. Right. So what is it going to lose?
It's gonna lose oxygen.
No,
No, wait.
Salt.
Yeah.
Salt. Okay. And when it does, what's going to happen to the shape of that cell?
It's gonna shrivel up.
What'd you say?
Right.
Just like the lettuce. I mean, the celery or whatever.
Yeah. It's the same thing. So it's going to shrivel up. What's going to happen if we actually, no, it's not going to shrivel up
You're not gonna shrivel up. It ain't no-
because what it gained.
They gained water.
Oh, so
I
it's
saw
gonna
it.
pop. It's gonna,
Oh, I was like on a roll. I was like, oh yeah, oh no, because look, it goes, and remember on the little video yesterday,
It can
pop.
and it explodes. Now, why would this not explode?
Because
they already had water.
it's hydrated. It didn't have water to begin with.
It did,
Oh, it did.
and if I keep putting water, what does it have that? And this was one of your questions on your lab. What do the plant cells have that our animal cells don't have?
so
So
Right. If we had cell walls, they wouldn't burst, but they do. So I mean, they don't. So it's going to do this.
Was that
So it's
better?
going to get so big
I wish I had a solo.
that it explodes. So it ruptures or busts open. Okay. All right. So it swells and it gets bigger. All right. Next picture.
hold on. You go something?
All right. So the next one is a box. It's similar to this right here. Just so that way I can show you kind of how you'll be able to use this in the same concept.
So,
Okay. So do this. Here's a membrane. All right, here's our cell membrane. Bojangles iced tea.
Oh man.
Okay,
Oh, that'd be
here we
good,
go.
man.
oh, is that your good?
All
right.
My girlfriend this weekend to go get the Bojangles. Yeah.
I walked in.
No, it's all right.
Okay, now this time we are not drawing dots. We'll use percentages. The reason is because they will put that on your exam. So I'm going to this is what we're going to do. I've got a box and I put a Ziploc baggie right through the middle. Ziploc baggies are membranes. They have holes in them.
Oh yeah,
They're tiny holes, but they have holes. So we're going to put a Ziploc baggie membrane right through the middle, kind of like saran wrap. And on one side, we're going to pour Bojangles iced tea without any ice. Okay. So it is 80%, and I'm really exaggerating, sucrose. All right. Sucrose is what?
That little mix you put into.
That's like sugar.
Sugar.
Oh, sugar.
All right. And we know it's tea also, but that means what percent water?
20.
Okay. Next. All right. So here's our Bojangles iced tea without water. Now, let's say that I say, okay, I'm going to put some of that 80-20 and I'm going to fill it full of ice and I'm going to let it melt all day. And I pour it on that side. What's it going to taste like?
Water.
Yeah. So it's 20% sugar and 80% water. So tell me what's going to happen. Yes, you do. High to low, go with the flow.
It's gonna even out.
Okay, so what's first going to happen? Or what's going to move in what direction? Choose one. Choose water or the sucrose. I don't
They're
care.
gonna go like against each other.
20% finna go to the left, go to the right.
All right, so where is the sugar going to go?
Right. The sugar's gonna go with sucrose.
High to the low, go with the flow. Right, so sucrose goes that way. Just draw an arrow from sucrose. All right, so sugar is going to move that way. What's going to move in the opposite way? By osmosis, water. So we're going to say water because high to low, go with the flow. So water's going to move that way until, Zayden, what'd you say is going to happen?
It evens out.
Right. So you remember yesterday they used the words hypertonic, hypotonic.
Yes.
Mhm. Which of these is hypertonic? Because it has a whole lot of solutes in it. Is it side A or B?
A.
Right.
Okay.
Because it's sugary, it has a whole bunch.
Yeah.
So this is hypertonic.
Yeah, I'd rather have some.
What does hypertonic mean again?
So
Yeah.
hyper means
Ew.
a lot
Non-sugar.
of solute or solute, which is in this case sugar, but it could be salt. It could be whatever you're dissolving in it. Okay. Hyper. So what do you call this side that's all watery, nasty kind of tea? Hypo. Good. Now what Zayden just mentioned was it always going to try to get a balance. The balance is isotonic or equilibrium. Okay, so eventually what we're going to have is, without drawing, I'm just going to draw a box and write, tell me what percentage to put. It's all going to even out. What's it going to even out to?
This YouTube
How many
is
funny?
gonna have a whole lot of ads.
50.
Oh.
It's gonna have a whole lot of ads.
What? Oh, yeah.
So I'll just put 50%. Be 50% on both sides, and that is equilibrium. So they say what causes us, what decides what moves? It's the concentration gradient. It's the difference. A gradient's just a difference. So it's the difference in two sides. You always compare, or isotonic, which is equal. Yes.
Okay.
And put that down because, I say equals, I put equal, put same. Same on both sides or something like that. Something that helps you. Because if you just copy my notes and it doesn't, you look at it, you're like, I don't know what this means. You got to put down something that'll help you remember it.
I get it.
Okay, so that's equilibrium. At that point, check this out. The molecules don't stop all of a sudden. It's like, oh, I don't move anymore. Now they move back equally. So you put a double-headed arrow to show that they're still moving, but both sides are the same. Okay? Does that make sense? Better?
Yeah.
Because yesterday it was one of those, oh my gosh, this is a Monday, and it's like, oh, it's not going in.
Yeah, you just gotta go over that a lot.
But this, if the more practice you get, the better you get at it. All right, last one that I know you can see on the exam is this one.
Sorry about your throat there.
All right, we're going to take a tube and we're going to bend it. We're going to put a membrane right in the center, just like
Magnet.
it kind of does look like that. This is actually this little tube. Anyway, that's a membrane. All right, we're going to do the same kind of deal. On both sides, on one side, I'm going to pour really salty water, 60%. Way saltier than what you're going to use in the lab. Today, it's like 10%, 5%. All right, on this side is 4%, 40%.
That's the hypertonic.
So which one has a higher concentration? The left, A or B? Let's do A or B again.
A.
All right, A. Now, tell me which way is the salt going to move?
To the middle. Oh,
No, to the right.
To the right. High salt to low salt.
Yeah.
Salt goes that way. What's going to pass it on the other way?
40%. B.
What's in the salt? What's in the solution? It's going to pass it. If salt goes one way, what's
Water.
water?
Osmosis.
Okay, osmosis. So this column will rise and this one will fall
Wait.
until it's all equal.
Oh, okay.
And then it's 50-50. It's kind of cool. All right. Everybody good with that? Kind of?
Yeah, I got it.
Okay. So go to the active side and we're going to draw a picture with the box to show active. Okay. So if this is go with the flow, that's what the lab is based upon. This lab is, you've got 3 sailors. They're all stranded on a desert island, deserted island. And one has found fresh water to drink. One has found a little bit kind of salty water to drink. And one has found straight up ocean water to drink. And you're going to find out what happens to the cellular cells in 24 hours. So you're going to set up the lab today, let it run for 24 hours instead of 30 minutes like we've done in the past. And we're going to really see what kind of changes happens. It's going to be pretty remarkable. So we're going to do osmosis diffusion in the lab, not active. So what's your question?
Does that say salt on the bottom?
Yeah. All right. So up here on the active side, you already said it's low to high. We need to add against the concentration gradient, against the flow. We're almost done with notes, y'all. So yeah, concentration gradient against the flow. All right, it's kind of like, no, don't go with the flow. That's what this one is. It's kind of, we have to use energy to be able to push it against. So the best way I can describe it is if you sit your butt at the top of a slide and you just, because of gravity, and you just kind of give yourself a little bit of a push, you throw your feet up in the air and don't let your bare legs hit the slide. What's going to happen?
Oh my God.
What'd you say?
What's going to happen if your feet are in the air and you just barely push off? What's going to happen to your body?
You're gonna go down.
You're just going to go down. You don't have to use your arms and like do this because your feet are up in the air, your butt's, back to where you can just slide on down. That's this. Go with the flow. High to low, go with the flow. Over here is like this.
Running up the slide.
Exactly. So we're going to draw a slide like this. So here's your high to low. Here's this person trying to climb up. And you need,
Thank you.
You need ATP to do it. So active, it takes a heck of a lot more energy to walk up that slide. So in terms of a box and with pictures of molecules, we're going to draw another box like this.
I'm gonna run up this
Right,
slide.
so all they were on the slide. So
Oh,
all
yeah,
they
that would be too.
just did, they were negative. No.
They were all passive, which will be high to low. So you'll be going, and if I drew another slide, I would say somebody sitting at the top with their feet up in the air and they're like sliding down. So tell me how I can do just with dots. So we're just going to do one set of dots. Which way will these go if it's active transport? High to low or low to
active
high?
transport, that's all the way down.
High
That's
to
high to low.
So should the arrow mean go to the left or the right?
Go to the left.
Correct.
Because
Low.
it accidentally goes against the.
You're exactly right.
How you know if it's active or not? Got one?
Well,
that goes out
and
less.
it always go from where there's, if the arrow's drawn from where there's less to where there's more, it's always active.
Okay.
So on your Ed puzzle, it showed how these special proteins act as pumps. You remember that? Did y'all get that far?
Yes.
And so it uses transport pumps, these transport proteins, to actually pump them because they won't go that way unless they're pushed. And you always need ATP. Otherwise, they're just going to keep sneaking back this way. And you're like, oh no. So pretend this is somebody who's trying to sneak out of their house and the parent is going, oh no, and pulls them back.
You got caught.
I ain't never get put back in though.
You will if it's active transport. So use this transport. So picture the pump will be your parents. So here they are standing here. Here's Zayden and Ebony, and they're standing like this, and you're trying to sneak out. And they're like, oh no, and they're going to pull you back this way. And it's going to take a lot more energy to do that. So,
Oh man, be yellow.
yes, okay. All right. So let's look at your homework paper and practice on each. And then after that, we'll look at your lab paper, which is also in there. And you'll set it up. Okay, so we have, I know you're getting up, I promise. And this,
So we done with the notebook?
yeah. All right, so you're gonna do 2 of these by yourself, real simple. We're gonna do one up, we're gonna do the first one. And then you'll do 2 of these by yourself, and we're gonna do one of them, okay? So take your colored pencil or your, I mean, it doesn't matter, I'm gonna use colored pencil still. I need to know if you can't read those percentages, because it's kind of hard and smeared.
80%.
I can't read
I
at
can't.
all.
All right, this one is 60. And that one's 70, 30, 60, 40. Okay? 60, 70, and then 40, 30. How about these? Can you read these over here?
Yeah.
Okay. All right,
You
so
can read
let's,
the bottom, then you can read the top, and it evens out to 60% glucose and 40% water.
right. Now this tells you to, I don't know, color stuff. We're not doing that. So all we're going to do is draw arrows. So where do you have more glucose? On the left or the right? Where's the number higher?
But
All right. 70 is higher than 60, right. So which way is the glucose going to go?
on the left.
All right, so you draw an arrow that way. All right, which way is the water going to flow by osmosis?
To the right. It's always going to be opposite.
That's all you do.
Okay, let me
Okay, so if it's, remember, if it's equilibrium, it's a double-headed arrow. All right, let's go down. We did the bottom left one. All right, look at these percentages and see if any of those you can't read.
40 to 60, 20, 50.
40, 60, 20, 50, yeah.
And
What about the
5 and
last one? The
25.
last one is 10-10.
No, it's not. It's 5 and 25.
No, I'm talking about
Oh, the other one
one is just the one, yeah.
Well, this one is actually 5 and 95.
No, go
is
up.
95.
Mm-hmm.
Oh
No,
yeah,
go
I
up,
see it.
this one. They're the same numbers. So
Right. Right. Yep. Okay, so tell me where do you have more water?
You have more water with
Inside or outside?
Inside.
All right, so which way is it going to go?
outside.
Out.
All right, so draw an arrow from the word water out by osmosis. All right, what's it going to gain?
So.
If it gains salt, what's going to happen to this cell?
It's gonna explode. I mean, it's going
It
hydrate.
lost water. It lost water.
So it's gonna shrivel up.
That ain't me.
All right, so you're going to say shrivel.
Shrivel.
How you spell shrivel?
Hey,
That's
SHRIVA.
Oh, yeah. All right.
hey.
And so it's going to look like
You got
this.
a shrivel.
I usually say shrinks because it's easier to spell. All right, so that's it. And all you have to do, if it swells, if it stays the same, you put that on the blank. That's all you do. Okay?
what was the word for ice?
Isotonic, or you say equilibrium.
I like equilibrium. That
Yeah.
should be equal.
All right, are y'all good?
Yeah, what's this? 83 or 80?
That's
Oh,
80.
Oh, never mind.
80,
That's
20.
isotonic.
What can I
Wait, what did you do under
We gotta work by ourselves?
So this is homework. You're just gonna finish that with homework and
Oh,
then
I
puzzle.
got you.
We're gonna flip to the lab sheet right now.
Alright.
So technically what we did was we already did that. Now we're down to here.
Factory.
What time is it? Okay. Oh, perfect timing. It took them 20 minutes to set up this lab. Probably give or take. Because you're smaller class, it won't take you as long, but all them having to wait on each other and all that. So this is perfect timing. Flip.
But the first one right here be it will get bigger because it got more water coming in. And less salt.
If there's more water coming in, it's always
Potato
worse.
core lab?
Yeah. Yep, potato core lab. So I did not run off the instruction sheet. I decided that I was going to make it simpler, and then I didn't run it off in time. I'll be very honest. So I just coached them through it, and they did a great job, and you'll do the same and setting it up, because we're going to run it 24 hours and not 30 minutes like it says. Okay? So the first thing we're going to do, because we're going to experiment the effect of salt water on somebody's cells. Instead of putting people in there or like cutting off some of your skin or something, we're using potatoes. Okay? Those are the little bodies of the sailors. So I've actually added this and made this more like an AP lab. So we're going to add, if you have a steady hand, you can add another section to it, or you can kind of use a straight edge. But we're going to go across like this. And you're going to have 2 experimental groups instead of 1. And you're going to be able to use some more equipment that we haven't used. We're going to use an electronic balance. We're going to use the graduated cylinders where you have to measure the liquids and put them into your tubes. I mean, so we're gonna use some other stuff that we haven't used yet. Okay, so we're gonna have 3 potato cores. Everybody's gonna get 3 out of this little container. Okay, so they're already pre-cut. They look like little chunky French fries. And you're gonna get 3 of them. One person in your group is in charge of this part. And it'll be right over here. So what you can't do is touch them because the oils on your skin will interfere and, like, you know how oil and water doesn't mix. It'll make a layer where the water can't move. So you have little tweezers or forceps that you're going to use.
Oh, man, I remember I used those when I was little. The forceps.
So much Here they are. Okay, so each group is going to get one of these. It doesn't have much in here. I've already made your tubes, okay, with the percentage. So the percentages match up with how much salt is in the water. There's a 0, a 10, and a 5, and that corresponds to the water over there. So let's put that on your lab sheet that's not on there already. So fresh water, what percentage of salt is it going to be in that?
0.
0. So let's put 0%.
Hey.
All right. Now we've added 2. So let's do 5% salt water, and we're going to do 10% salt water. This is where you're going to put the mass of the potato core in grams right here. So whoever's the potato core person who's going to pull them out, dab them off on the paper towel, and weigh them, you're going to put the masses right here. Okay? And then you'll give this information to your group. Tomorrow, it's going to be 24 hours, not 30 minutes. We're going to let them run, and we'll see what happens. I think you're going to get good data. And then we're going to see what kind of change might have come about. So which of these will be considered the control group? So we're investigating the effect of salt water on your cells. So what's the control? Salt water is going to be the special treatment.
So fresh water?
So, yeah, the one that's in the fresh water. So let's put 0% potato. Okay. What are the 2 experimental groups then? Who's left?
It's water.
Yeah. So 5 and 10% potatoes, saltwater potatoes. Okay. So what's the independent variable? What did we put in this water that's not in the control? Or what did I put into the water? Salt.
Salt.
All right, so we're going to put salt concentration because it's different, or percentage. And then what do we think we're measuring? What's going to happen? What are we measuring here? The mass. Yeah. So we're going to see what happens to the mass. They might gain some mass. They gain water. Some of them might lose water. We don't know. But you can predict what it's going to be. All right. So we have 4 groups set up. You definitely need 3 people in a group. Just because you need to be able to measure your liquids and be able to get that. Somebody needs to work on the potato. If you do have a group of 2, it's just a lot more for somebody to do. But I mean, it's possible. But I think groups of 3 would probably be the best. Okay. All right. So figure it out, and then come get your stuff, and you can start, y'all.
Hey, people. Want to be in my group? We can do this work. Get a good grade. All right. Let's group up. So,
Yeah, this is also a little test grade lab like class. Okay, here we go. Y'all, each person.
you want my present? Hey, baby.
Got 6-0 win. 58%. Yep.
Over
Yeah,
here.
it's all right.
To the base.
So you want to be in my group? My group.
I have a group. We need one more person.
Oh, you got to
I'm with Zayden.
Alright.
Yeah, you.
They number 1 in Navigation.
Right now.
You're
Yeah.
What is that?
One goal.
You can't get Jesse.
No, it was crap. The referee was calling us. They called him good 20 times.
We could get Ebony in our group.
Yep.
Yeah.
We don't need your color picture.
All right, y'all, come on. No, come on. But we gotta set this up. It takes a while, y'all. Come grab one of these, whoever, a representative from your group.
Hey, Teron, you with us, man? Hey, JD. Hey, JD. You with us, bro?
What is this? What is
It's a rodent.
You wanna live on the ground?
Alright, add me on Snell.
Stay with your group now. I have to put you aside. Tayron, who's in your group?
Back to the basics, back to the basics.
Y'all come grab one of these. Nick, your group. All right, will you ladies share data with Nick,
Anybody in school.
please?
Right.
All right, come on, Zayden.
Who group am I in? Who group I'm in? Who group I'm in?
Take that to find a spot. Doesn't matter. Y'all decide who's gonna do what. And Zayden,
Yeah.
here.
Oh, my group?
Okay. All right, everybody find your spot. JD, come on. Anyone in your group?
Bro, did you just touch my tooth?
For my group.
Okay. Y'all think that the potato weighing station. So whoever's gonna do the potatoes, you're gonna come here. Whoever's measuring the liquid, come over this way. It's only salt water.
Where the potatoes?
Alright, so, right,
You with us?
please grab them, please, 'cause I'll move.
You can't. JD's with us.
No, she was.
JD's already said he
I'm over here.
I told you.
Back to the basics, back to the basics.
Hey, y'all,
Stop.
come on, get with your group.
Calm down, bro.
Alright, let's go.
Okay,
Let's
so you have to
go.
come here.
Let's go.
So you're gonna measure it, 20
Now,
milliliters.
hey, you gotta leave from our group area, please.
So 20 milliliters. The rest can only
Yeah,
say.
this way.
The yellows are this
And
here,
I just put
and
it on
the liters
there.
for this one. Okay, so it has on here what percentage.
Ain't no
So you need your little container.
She said, 4 feet, make sure if we call her that, we gotta call her 4'6". Exactly. Ask you, hey, remember you said that?
Let's get.
No, 4 foot.
4 foot? My name's 4.
Wisdom 17.
Let's go get this stuff done. We got it.
17 feet.
So what
Let's
you're
go.
gonna do.
Everybody get one of these.
17
Here you go.
foot.
Here, you get one.
5%.
Back to the basics,
10%.
back to the basics.
You get the 10% milk, you get the 5%, I'll get ready.
You have to let some off, like you have to let the extra water.
Come on.
Mine says 3 times.
So that's potatoes. All right, whoever's measuring their liquids, it takes a while. You need to bring your tubes. Come over here. Yes.
That's 5%,
I gotta get 5%.
that's 10.
All right, so
This is-
you measure 20 meals. So that means 20 meals.
It's 20 drops.
No. Right.
What 10% is
There's 10 meals.
Oh, you gotta add those to it.
Right. Now, you can pour it. It's a little off. You can pour it, and then you can use that to help you lift it up. If it goes over, you trash off. You never add it back into it. You need to put the right here. 10 mils should be here at the very top.
Oh, 10.
Well, you gotta do it twice. Okay. All right, hold up. You can't mix them up. So 10 has to be with these reds. So you can come around the side, yellows, and then the clears or whatever this one is. Okay. Send somebody to do your potato, but it's measuring. That's why I would pour it and then use that to help you trash off it.
When I first got it, like little ball.
I know.
Oh yeah, this is right on.
Ooh, I'm trying to
20 mils in each. It's just you can't mix up these.
I want nothing now.
Medium and that's.
I just poured it here now.
I think it's on here. Make sure you put it in the right thing because that's the 10% goes in the can. Yep.
What are we doing?
Yeah. All right, so you already done it?
Yeah.
All right, so what did you put them on here? Do you know which one's which?
This one?
All right, but how much did it back weigh? You gotta record it on your paper.
My measurements be on point. You see this though, you see this right there. Spot on.
3.6, man.
Okay, so write that on the paper. Yeah.
My daddy knows that.
Hey man, you freaking.
Okay.
That's
Now,
good.
right there.
Rub this button on.
And then what you're doing, Solomon, is with the 3.6 grams.
Back to the basics, back to the basics.
Huh?
Back to please, you gotta tell.
Alrighty. I'm a fish. I'm a bass.
These yellow
Watch me
stay
do
with
this.
the distilled,
Raw.
those stay with that, and the red stay with this. Of each solution.
Freaking in my way, man.
That's the zero.
I need some taters, please.
I got some of this in there.
Would you like to beat my taters?
Back to the basics, back to the basics, back to the basics.
Oh, God.
Hey.
Why are you asking me? Now, all you do is you take this back over there and you'll drop them into your, and measure the water.
Back to the basics, back to the basics.
Take it back to your station and you'll put the
You don't even like me, bro.
Yeah.
Put them back into your 5 and 2. And people know what it is.
But I knew what this
Alright, now
was.
you're-
I need some big tweezers. All I work with.
What are you thinking?
You take it back to your table and you're ready for the potatoes.
Hey, young boy.
Make it
Fine.
right.
Come on, what opposites do we have?
If I'm
5 and 10.
And you
Thinking
write it
I'm
on
gonna
paper.
call it. Whoever did 10% over here ain't doing good. JD.
We're using this just, yeah.
Yo, my-
You did 10%.
Yeah, I did 10%.
Fill it back up again. You gotta put 10 more milliliters in.
Actually, you
Huh?
know.
You gotta put 10 more milliliters in.
3.0.
You gotta put 20 milliliters.
Well, you get the 5 to
Okay,
10.
so what you wanna do?
0.
No, it's dead. It's dead.
Wait, who's that one?
No, not my phone.
Oh, no.
Here, make yours like this. Write this out.
Oh.
So the next one will be 5%.
Oh.
I gotta go. So that way you don't get mixed up. This,
Yeah, I hear that too.
That's
what is it?
oh, turn it off.
Y'all heard that?
You go to the, it's the thing
Okay,
on
and
there.
then make sure you put G for grain. Now you're ready, just transfer with your forces over to the water.
Oh, wait.
You ready?
Drum.
Yes.
Yeah.
Okay, come on over.
Can we use this? Can we pick these up?
I don't know.
This Thornhill. Do we need the clock? Do we need the little things?
Forces.
The little.
Yes.
I got 30.
You have to do that?
Sure.
No, you have one. There's
Hey,
one.
come on. For sure.
There's not one you need.
For sure.
Alright, so now, what's that? 2.4?
Back to the basics, back to the.
Okay, 2.4 G for grams.
And JD, what was the name of that song, bro?
Oh, you broadcasting?
What?
Zero viewers.
Okay, now, it doesn't matter.
Hey, JD. What you name of the
What are you
song was yesterday?
Which one?
The one I asked you yesterday.
I forgot.
Hey, Ron, are you
Which
good?
song did you ask me?
That's what I'm gonna think of.
Which song did you ask me?
I thought I knew you.
Grapevine, Great. Great Bond.
Go ahead and start packing my stuff.
I thought I knew you.
Just make sure you get the data. What time is it?
36.
All right, we're in good shape. You just need to get the data and then pack it up.
Nuts, boo.